Great. Well, looks like we've got 21 people on. I'm sure we're going to get some more people here today, but let's get started. I just want to thank everyone for coming and attending this webinar by folks out of our research arm of the U.S. Army Corps of Engineers. It's the U.S. Army Engineer Research and Development Center. This work was approved by the Middle Rio Grande Endangered Species Collaborative Program Executive Committee as being important work to help with adaptive management of the Rio Grande and specifically of the Rio Grande Silvery Minnow. And then it was funded through the Corps of Engineers Collaborative Program. So the Corps of Engineers receives its own funding to participate in the collaborative program. And our program funded this research. Just for those of you who may not know about or not familiar with the collaborative program, it is a group of 17 signatories. It includes federal agencies, state agencies, non-federal organizations, and nonprofits. And it was formed in 2002 to address concerns about water management on endangered species, which includes the Rio Grande Silvery Minnow, the southwestern willow flycatcher, the yellow-billed cuckoo, the Pecos sunflower, and the New Mexico meadow jumping mouse. And so this work will help, hopefully, the plan is to have this work help inform where these minnows are and inform how we can better sample for them, and then therefore understand where we need to focus our efforts for recovery, but also to avoid impacts of various activities such as removal of vegetation or removal of islands or any kind of earthwork within the river channel. So with that, I'm going to hand it over to, I think, Mick, if you haven't already. Sorry, I was a little late. Give it a little bit more in-depth introduction to the presentation.
Okay, well, it's my pleasure to introduce Dr. Richard Lance and Denise Lindsay. Dr. Lance is a senior research biologist with the Environmental Lab of the U.S. Army Engineer Research and Development Center in Vicksburg and team lead for environmental genetics reconnaissance. He's been working in the field of molecular ecology for nearly 30 years and in the field of environmental DNA since 2009. In 2014, he helped establish the government eDNA working group, and for the last 10 years, he's helped direct this informal collaborative effort that includes more than 100 eDNA scientists working with and for different government agencies. Denise has worked with Dr. Lance on the genetics reconnaissance team at ERDIC Environmental Lab for 18 years as a research biologist. She conducts investigations into genetic properties of threatened, endangered, and invasive species, utilizing the latest genetic analysis tools. Her field and laboratory efforts include population genetic studies, genetic survey methods for plants and animal DNA, as well as environmental DNA and RNA from water and soil samples. So with that, I'll turn it over to Denise.
Well, hello, everybody. I have got the presentation fully encompassing my screen, so I can't see any of you, or if a chat comes up or a hand goes up. But please know that I'm not doing this super formal, so if you have a question and want to interrupt, just go ahead and let Dr. Porter know, and he can jump on and ask the question. Or if you'd rather save it to the end, we'll certainly have time for that. So I'm going to forward the slide and let me know if everybody can see it, if it's working. We had a little bit of a lag earlier.
It's working.
It is working.
Fantastic. Okay, since I don't know everyone who's online today, just in case there's anyone who doesn't know what eDNA is, I thought I'd just give a quick little background. eDNA is just simply DNA that's shed into the environment. It can be shed into water, soil, or air, and then it's later collected for genetic analyses. So in regard to the Rio Grande Silvery Minnow, a fish, we're going to assume that their eDNA is generally derived from the mucus or the slime coat, feces that are left in the water, gametes that get into the water, or if the fish is injured and they shed DNA that way. So detection of DNA from our species of interest in an eDNA sample is indicating that the species is or was present in the location that we're sampling. And so for this project and for other eDNA marker development projects that our team has done, what we aim to deliver is 2 highly sensitive and efficient qPCR assays. And the reason that we want to develop 2 markers or assays is because using more than 1 marker increases your chances of detecting your target species, especially if that's a rare species or a small-bodied fish that may not be leaving a large quantity of DNA out in its environment. Using multiple markers also helps to alleviate the risk of non-detection that's due simply to PCR inhibition, because different markers react differently to inhibitors. So what are the benefits of using an eDNA tool over traditional survey methods? So for us, it's mostly that you can cover more ground more quickly. So you're having to spend less time and labor in the field. When you're out there collecting samples, you can get to more places a lot faster than you can if you're having to seine for fish or electrofish or other ways that people do observational studies. Additionally, eDNA surveys require no taxonomic identification expertise, so any technician can collect the samples. After the samples are collected, at this point, we're still requiring labs with genetic expertise to complete the genetic analyses, but the turnaround time and the cost for those things at these labs, like our lab, are decreasing. Every year they decrease, and currently, there are multiple field portable genetic analyses machines that are coming online and are becoming more user-friendly and able to be used for different types of DNA, and including eDNA. Another benefit of eDNA sampling is that it is non-invasive. So if you're working with a protected endangered species, you're not disrupting that species in order to find out whether you are detecting it in that location or not. And then finally, the eDNA tools that we develop can detect very low amounts of DNA, of the target DNA that's out in a sample. So even just a few copies of the nucleotides of the DNA that's out in a larger water sample, you have a higher chance of detecting that species than if you were simply looking. So to get the project rolling, we started with 2 different types of samples. The first was getting some high quality Rio Grande Silvery Minnow samples, and we obtained those in the form of tissue and eDNA. The tissue came from mort fish, fish that died in transport to ERDIC here for other studies that are going on at ERDIC, not that we're using. And then the eDNA came from holding tanks where they are keeping those fish. And then the other type of samples that I needed were non-target species. So in order to get fin clubs from these species that are listed here, we used a combination of collaboration with other labs that had samples available that they could send to us, that they had stored in their freezers, like tissue samples or fin clip samples. And then also Dr. Porter went out with his team on site and collected, via seining, he collected the different species that cohabitate with Rio Grande Silvery Minnow. So the goal of testing this Rio Grande Silvery Minnow eDNA tool against the non-target species that are present in the Middle Rio Grande, as well against non-target species that are related to the Rio Grande Silvery Minnow. So that's why we wanted to get some of these samples that weren't available there where Dr. Porter was sampling. So initially, we had planned to design our eDNA markers from new sequence data that we were obtaining from the mort fish that we collected. We ran 11 individual Rio Grande Silvery Minnow samples through long-range PCR and high-throughput sequencing to obtain new whole mitogenomes. And that sequence data aligned pretty well, though not perfectly, with the 4 available Rio Grande Silvery Minnow mitogenomes that are already available in GenBank. For anyone not familiar, GenBank is a public database of nucleotide sequences that's managed by the National Center for Biotechnology Information. So our next step then was to align our Rio Grande Silvery Minnow metagenomes against the non-target species. And to do this, we included sequence data that we generated from the fin clip and tissue that we obtained from Dr. Porter and other labs, which we sequenced alongside the Rio Grande Silvery Minnow. And then we also pulled metagenome data that was available in GenBank. So here we were able to identify conserved loci and then look for regions of sequence that had enough nucleotide differentiation, that would be sufficient to prevent amplification of any of these non-target species. Meaning we wanted to design primers that would only amplify Rio Grande Silvery Minnow DNA and nothing else. This was done as in silico testing using the software Primer Blast. And the reason that we do that to start with is because it narrows down the number of draft markers that are worth investigating in vitro. So to find the loci that are target specific, we look for sequence mismatches within the primer set, and the primer set being the forward and the reverse primers along with the probe. So the more mismatches that we find, the better we are. They need to be present on both the forward and the reverse primers, and they need to be present in the probe. So an example that I'm showing you here on this slide is looking at one of our primer sets against one of the non-target species, and you can see that there are 5 to 6 mismatches, nucleotide mismatches between each primer and probe with that non-target species DNA sequence. The primer sets that we design are qPCR assays, and that means they return our results in real time, quantitatively, as the PCR cycles are completed. And the reason that we use probe-based qPCR assays in our lab is because they are highly sensitive, because the probe increases specificity of the assay, and it makes it even less likely that non-target DNA would amplify with the assay. So for our initial testing of our 29 draft markers, we run traditional PCR rather than qPCR. And that basically just saves us the purchase of expensive probes until we are ready to test assays that prove that they are accurate and reliable. So our first reference genome was created from a consensus sequence of the 11 mitogenomes that I developed. But those assays proved to be non-specific to Rio Grande Silvery Minnow despite multiple optimization attempts. You can see in the top gel here, it's showing just a subset of the unsuccessful markers from that first reference that we used. The samples listed in green are the Rio Grande Silvery Minnow samples, and the samples in black are the non-target samples. So you can see we had a good bit of non-target amplification in each of those initial draft markers that we were looking at, and then alongside them, the Rio Grande Silvery Minnow was not amplifying cleanly. So there's multiple bands in the gel. So we knew something was up with all of those assays, and we ended up dropping them and going back to the drawing board. And we employed a new reference sequence to pull design primers from, and that was a GenBank metagenome that was already available, one of those 4. So these new assays, 5 of them, 5 draft markers, amplified only Rio Grande Silvery Minnow during traditional PCR. And looking at the bottom gel, you can see how clean it is. We only see a single band for all of the Rio Grande Silvery Minnow samples that are there in green. And then the non-target samples labeled in black, there are no bands. So all 5 of these assays were worth moving forward to the next round of testing, which is qPCR. So for qPCR testing, we run samples in triplicate, and then we optimize to increase specificity. So for our 5 draft markers, we had 2 that were in the ND1 gene, 1 that was in ND4, 1 in ND6, and 1 in the COX1 gene. Our COX1 assay amplified most of the non-target species in qPCR, despite it being clean in traditional PCR runs. So that assay was dropped from further analyses. And then the 2 ND1 and 1 ND6 assays had 0 non-target amplification, meaning that none of the triplicate replicates amplified any of the non-target species. Additionally, both of the ND1 markers and the ND6 assays consistently amplified all replicates of the Rio Grande Silvery Minnow tissue and the Rio Grande Silvery Minnow eDNA positive controls. So we just wanted to note quick that we had one sample of one species that had amplified with all the assays, and it was likely due to contamination, but that can happen at any step during the process. And all we had to do was raise the annealing temperature by a couple of degrees, and it eliminated the problem with that one single sample, and it did not compromise the efficiency of either of the ND1 assays or the ND6 assay. The reason that we will not be moving forward with all three of them, though, is because the second ND1 assay has primers that overlap a bit with the first. And so they wouldn't be able to be used in multiplex if that was something that folks wanted to do down the road. And we chose just one of the ND1s and then one ND6 marker to move forward with for field testing. So once our assays had been validated, it was time to head into the field. So Dr. Porter headed out with his team, and they collected water samples from 10 different locations, and those included sites where the Rio Grande Silvery Minnow is known to occur and presumed to be absent. Those samples came to me labeled in a randomized number scheme to prevent analysis bias on my end. I didn't know where they were collected. I didn't know what any of the codes meant. Everything was numbered sequentially, but not by location. So they were essentially blind samples to me. So I ran them without knowing what to expect while looking at them. Also, when the samples came to me, they were stored in bags of 10, and I just extracted each bag of 10 by itself. And the reason that we do that with an extraction blank is to ensure there's no cross-contamination between samples or between sites. So to collect the samples, water was filtered with a single-use 3-micron filter that's fitted into a single-use filter cup, and it's attached to a peristaltic pump. So you can see in the picture here, the filter cup is placed into the flow of water, and then water is pumped through that filter cup over the filter, capturing eDNA on the filter, and then the sample is collected after you've reached a certain amount of time or volume of water, depending on what your goals were. So turbidity often differs by site where you're sampling, and it can cause those filters then to clog at different volumes of water. So we just aimed to have Dr. Porter collect as close to 1 liter of water as possible at each site. For collections all the way through, sterile technique is really important when you're working with eDNA in order to prevent cross-contamination. So single-use gloves are used, single-use tweezers are used to move those filters from the filter cup into a storage tube. Every sample has its own tube, and those tubes are filled with desiccant to dry out the filter a bit and preserve the DNA. And then those desiccated filters are stored in the refrigerator just at 4 degrees Celsius until it's time for me to extract them. Like I said, with the way that the samples were separated into the different bags, I ran 10 different sets of extractions so that each bag was extracted at a different time and didn't overlap. I used QIAGEN's DN-Easy blood and tissue kit, which is very standard to use. However, we do modify for eDNA sample extractions to add 3 initial bead beating steps, and this just helps the lysis buffer to break down the filter itself, as well as the eDNA that's on the filter. And it causes us to increase different buffer volumes for the first few steps of it. But then other than that, we just follow the protocol all the way through to the end. But I will note that when I sampled the holding tank of Rio Grande Silvery Minnow here at Erdic, those were not filter samples. Those were just water grab samples, just for ease of collecting them quickly. And so rather than using the bead beating step, I pelleted those tubes in a centrifuge and then moved on from there following the same extraction method as I did for the filters. All the samples were run in triplicate for both the ND1 and the ND6 assays. And this also included all of the field blanks along with the field samples. And additionally, positive controls for the Rio Grande Silvery Minnow tissue and the holding tank eDNA positive controls. I also ran in triplicate all the extraction blanks and then qPCR also includes no template controls. So instead of putting template in, you just put in sterile water. So while blanks are used in all genetic analyses to rule out cross-contamination, it's especially important with eDNA to ensure that our amplification results are reliable. When I plated the samples then for qPCR, I purposely placed them back into sequential order rather than by bag number or by group number. And this would, down the road, reduce concern over a splash contamination. These plates have very small wells. There's 384 wells per plate. So you have to be very careful when you are adding your template to each well. If you look at the depiction of the plate at the bottom there, you can see there's grayed out areas. So the ones that are kind of intermixed are the field blanks in there. And then at the bottom, I have the extraction blanks, and I intersperse some of the no template controls with the positive control tissue samples. And that just helps us to show that as long as the blanks are all blank, then we can trust everything else that's amplifying in there. So on this slide for the ND1 assay, we're showing expected amplification patterns. The Rio Grande Silvery Minnow tissue positive controls amplified well ahead of the eDNA positive controls. The earlier the cycle of PCR that the amplification plot crosses that threshold line is a greater amount of target DNA that's present in the sample. So you may also notice on that bottom graph of the field eDNA samples that there is a wider range of cycle threshold values than there are for the Rio Grande Silvery Minnow eDNA positive controls. Some of them amplify earlier, some later, and that's not unexpected. Template DNA amount varies greatly by sample based on sampling method and the size of the water body, the amount of water that you filter, the degree of mixing and water flow in that water body, water quality, presence of inhibitors, and other things. And in the very bottom right-hand corner of that amplification plot, you can see samples that failed to cross the threshold line before the 40 cycle cutoff. And those are samples that we say were not detected. So those samples then did not contain Rio Grande Silvery Minnow DNA. We see a very similar pattern for the ND6 assay. Both assays provided 100% amplification of positive controls in triplicate, meaning every replicate of the positive controls amplified. For the negative controls, including extraction blanks, no template controls, they didn't amplify with either assay, and that provides evidence of sterile methods from sampling through to qPCR. And only 2 of the 10 field blanks amplified, and those 2 that amplified only had 1 of the triplicate replicates amplify. So that's just minor amplification. Once my lab work was complete, I reorganized all the samples by the 2-letter group designation that had been provided in the sample collection data and sent that off to Dr. Porter to see what he thought. I determined the number and percentage of amplifications per group, the average number of replicates that amplified per sample from the triplicate qPCRs, and the average cycle threshold value for each group. What I saw was that group RG showed the highest percentage of amplification with about 80% detection, while the other groups resulted in 10 to 25%. This didn't mean anything to me until I consulted with Dr. Porter and found out what the group designations meant and the location that each of those samples was collected. So to remove the blanks and the positive controls, this table simplifies the field samples, and it provides kind of an overall context as to where the samples were collected and whether Rio Grande Silvery Minnow were expected to be detected there or not. So in the Rio Chama, the Rio Grande Silvery Minnow was presumed extirpated, and we had only 1 replicate of 1 sample amplify with both assays. In the irrigation drains, which is a place where the Rio Grande Silvery Minnow is presumed present, we had 1 to 2 replicates of 2 samples amplify with both assays. In the mainstream Middle Rio Grande, where we know the Rio Grande Silvery Minnow is present, we had 2 to 3 replicates of those triplicate qPCRs amplify 36 samples with ND1 assay and 35 samples with the ND2 assay. And then in Tingley Pond, where Rio Grande Silvery Minnow is presumed to be absent, we had 1 to 2 replicates of 4 samples amplify with ND1 and 2 to 3 replicates of 5 samples amplify with ND6. So we can look at that even a little more closely, looking at each result by the exact sampling location, where approximately 10 samples were collected at each site, with one of them being the field blank. And we can see exactly where these detections were occurring. So the Rio Chama and the irrigation drains had very minor detection, just 1 replicate of 1 to 2 samples that amplified. So it is pretty unlikely that Rio Grande Silvery Minnow were present there. Sometimes you get a little splash over in qPCR. All of the rest of our blanks were clear, but it's something we can look into. All the samples from the bridges in the Middle Rio Grande, where Rio Grande Silvery Minnow are known to be present, had very high detection rates, between 78 and 100% detection. Samples from the Buckman diversion, though, where the Rio Grande Silvery Minnow is presumed extirpated, had a 44% detection rate. We had 4 of 9 samples amplify with both assays. And we think that this is a site that would be worth revisiting for both traditional sampling and eDNA sampling, and for which we are planning to sequence the qPCR amplicons to confirm that it's Rio Grande Silvery Minnow DNA. And then finally, for Tingley Pond, where Rio Grande Silvery Minnow is presumed absent, those 13 to 33% detection rates were only concerning until we learned that the irrigation drains where Rio Grande Silvery Minnow is suspected to be present feed into those ponds. So in this scenario, Rio Grande Silvery Minnow DNA could be accumulating in the ponds that don't have an outflow, or maybe not a regular outflow, because DNA has been shown to persist in the environment for weeks or sometimes months. And even degraded DNA can be detected with a highly sensitive qPCR assay. So to assess the sensitivity and the efficiency of our 2 assays, I ran synthetic Rio Grande Silvery Minnow DNA in a dilution series, and this was to determine the limit of detection and the limit of quantification for each assay. The LOD is the lowest concentration of template DNA that produces at least 95% positive replicates. And so what that means is we'll get reliable detections at that LOD concentration, but it's still possible to get detections at even lower concentrations. And the LOQ, the limit of quantification, is the lowest concentration at which template DNA can be quantified with a coefficient of variation value below 35%. So note that LOQ is always higher than LOD, but both metrics are used to compare assays so that other researchers can determine which ones are the best to use for their investigations. For eDNA researchers, we're most interested in detections versus non-detections. So when multiple assays are available for a species of interest, their sensitivity and efficiency metrics just help us decide which ones are going to give us the highest likelihood of reliable detection for that species. All the metrics for both our assays are well within the range of expectations for good eDNA markers, with the ND1 assay being slightly more sensitive than the ND6 assay. To close, I'd like to restate that it's important to use more than one assay in eDNA investigations. Confirmation of a sample's amplification across more than one assay leads to much more reliable results. And while we developed these assays for tandem qPCR, that is running each assay separately, you could use different fluorescent dyes in the probes and multiplex the assays. You would just need to do a little bit of optimization. And multiplexing is something that would be especially useful if the eDNA tool is ever transitioned to a field portable qPCR machine with in-field DNA extractions, so that you could receive on-site information while you're out there, whether you have detection or non-detection, while you're still finishing up field sampling. Every year, this technology improves as far as reliability and cost and ease of use, and it could be advantageous for genetic monitoring of endangered species. And that's all I have. So I'm ready to answer any questions.
Great, thank you so much, Denise. So this was, I think, really exciting information about not only the utility of eDNA and locating silvery minnow, but also on potential areas where silvery minnow could occur, and helps us thinking about what are our next steps, where else would we like to sample. So as Denise said, we're now going to open up the floor for any questions or discussions. I see, Lynette, that you have your hand up. You want to unmute yourself and ask a question?
Hi, great presentation. It was very interesting. I'm curious about the tingly ponds, just because it was my understanding that they only received groundwater. So I'm wondering if you could expand on which ponds were sampled, and if you would confirm with the biopark that they are indeed receiving ditch water, because when we reconstructed them, there wasn't a mechanism to take water from the ditches that I remember.
Okay, I'll have to let Dr. Porter answer that one.
Right. Okay. So I thought that the water source was from the irrigation drain. I might have been mistaken on that. We don't have a good explanation as to why we were sampling in the second pond. So you've got the sailboat pond, and then the next pond is the fishing pond, and we were taking our water samples from there. So we don't know why we detected silvery eDNA in there from silvery minnow. We think we need to
bait.
Well, that's entirely possible, or some bird happened to drop one in there. We just don't know.
Interesting. Okay.
Casey?
Thank you. Great presentation. My question is more about what's the practicality of using this type of species identification and when it's in a water body that is in constant motion, meaning, yes, you've identified that a species is present, but how can you be certain about where that species is in that environment, given that the sample you're sampling is in constant motion? I guess you could sample at multiple locations at the same time within the river to try and track that down, but it seems useful, but only to a point. Do you think there's a way to get around that issue?
You're correct in that DNA moves along with the water, but it does also settle out, and it can accumulate in the sediment at the bottom of the water column. And my understanding is that some of this area is pretty shallow, and then simply the flow of the water back over that area is going to disturb anything that settled on the bottom and kick it up again. Researchers that are out there collecting these samples are going to disturb the sediment and knock some DNA loose, knock it around to be collected. When you are getting detections above a point where you would expect to see these fish, that's when you'd want to go back out and resample, both with traditional and the eDNA methods, to see whether or not your detections are true, because you could get maybe false detections further down as the water is flowing down, but you wouldn't expect to see that up above.
I would say that because
Rick is in here with me.
Yeah, I would say that since the eDNA is, it's dispersed in the water column, it settles and it decays, then unless you have a really large population upstream, your source is probably fairly close to where you detect it. I don't know if you want
that my follow-up on that was if there's been any research that documents kind of the rate of settling, related to a known velocity. A lot of the habitat samples that we're interested in sampling are very low velocity areas. So I do tend to agree with you that if you're collecting DNA in that area and you have low velocities, you're probably finding DNA from species that are in that area. But I'd be interested to hear if there's been any research on trying to correlate the DNA found, related to any known velocities of or rate of flow. Thanks.
Yes.
Yes. You can find that information in multiple papers, but it's specific to the area that you're sampling. So if you really wanted to know for your species of interest, you would have to do some validation on your own.
Right.
Thank you.
And it also is related to the species, right, Denise, in terms of
Yes.
how much DNA it sheds, how big they are, all kinds
How
of
much they move,
Even the type
right.
of affected.
or
So-
even the type of substrate that's below the water can make a difference.
So, and I've seen other papers where you would do this effort here where we're looking at eDNA and how it's useful to determine the presence or absence of a species and also maybe the abundance. And it has to often be checked with actual real sampling in the field before you can start using it in areas where you don't necessarily know if they're there or not without doing the actual capture of the species.
Yeah.
So initially, we're gonna have to correlate it with field data, and so that's I think one of the things that was really interesting was areas upstream of Cochiti, right? And so now we are going to go out in the field, and there will be efforts to go see if we can capture some silvery minnow in those areas, like around Buckman diversion, where there were positives, where we had positive eDNA samples, and see if we can actually find the fish. So
Yeah.
This is part of a larger effort, right? We're always going to have to do some element of sampling, either in the front end or to verify what we're finding in the lab.
Yes.
Right. It's really standard, and we find that for all eDNA studies, you really do need to have a ecologist, someone that is an expert on that target on your species of interest, that really knows a lot about their life history and their movements, and the best time of year to sample, and how weather may affect it. Like, there's so many factors that it's really important to try and correlate all those different conflicting factors in there.
And at the start, using it just to identify places to look harder is really.
Yeah. So using this eDNA tool as here's where we saw something interesting. Let's go double check. Let's look a little bit harder. That's a great use for the tool also.
I like that idea also, as you were mentioning, is if you're sampling in a systematic way, you can say, well, there's some fish between this upstream point and this downstream point. But then as you go upstream, if you have a negative, then you can feel more confident about the downstream positive as being more localized, as being from that area. So just having sort of a systematic sampling effort can help you hone in on where they are to get a better idea of locality and really connecting those eDNA to a specific area. I see there's some information in the chat. I had put in a link to a paper about using eDNA for abundance studies. And then Thomas Archdeacon also included a link to a paper on fish data from the Buckman diversion area. Was there any other questions or comments? Oops. Looks like someone got bumped off. Here. I'm just, Debbie Lee.
Yes, I had a question about the applications for using eDNA for terrestrial species. So this has been about fish, but is there any potential applications for using this to look for, say, the New Mexico jumping mouse, which we are at such low numbers on that this would be very useful to find if there are other populations out there.
There's been jumping now.
There has been jumping mouse eDNA work done.
Okay.
I'm pretty sure.
I think they didn't find, I think I saw the abstract for a presentation. Oh gosh, was it at a Wildlife Society meeting? I forgot where it was, but I saw the abstract for it. And I think they did not find it to be useful in their study. And I didn't go any further to find the paper or see if there's a paper that they've written up on it, but they have started to explore using it, and it's used for all kinds of species. So they're using it. Some of the stuff that I think is really fascinating is the work that they're doing to look at pollinators. And so they go and they sample eDNA on flowers, and they can identify all the different types of pollinators that come to those flowers, and they could start connecting plant species with pollinators and looking at those relationships. And some of the things that we've talked about also is, like for example, for the Sacramento checker spot, which may be extinct, but going out in some of these areas where they were known to be just 2 years ago, and also swiping samples from different flowers and seeing if they're there and if we've missed them. So they can, this can be, this has a broad application.
We have
Great, thank you so
applications
much.
to connect pollinators and plants also.
Say that again?
I said we have done that using
Okay.
eDNA to collect plants and pollinators. Looking mostly at pollen collected off of petals.
Well,
I
the
think
And
it's really
bees.
interesting.
bees and pollinators. Yeah.
That's really great work. Any other questions or comments?
Andy's got a question.
Yeah, I got a question. I mean, you guys did your due diligence and trying your best not to contaminate samples and all of that. What do you suspect is the level of certainty, I guess, for the Buckman diversion samples? And would it make sense to go back there and redo eDNA samples before any fish sampling is done?
Okay, I don't have the Buckman broken down by the number of replicates of each of those triplicate qPCRs, but I can look at that and see whether it was just like one replicate was amplifying. But to see 4 different samples amplify, I think that's pretty. That's
And
pretty
actually
good.
it was 5 samples, 4 on one marker and 4 on the other. So there's one marker that didn't overlap. So there's actually 5.
Okay.
But no, I think it's probably worthwhile to go out and resample for eDNA again there. I was thinking about that myself, and yeah, I think that's a reasonable thing to do.
Yeah, just from a cost-effective standpoint, right?
Oh yeah, exactly.
Be a lot easier to sample water than send a group of people out there. But yeah, I'd be interested to see that area resampled and see what comes back.
Okay.
We have the markers and they're on hand ready to go. So I'm happy to run any samples that y'all send my way.
Great, thanks.
And Mick, yeah, like Mick said, he's thinking of a sampling design where we would sample for additional areas where we think that they don't occur, and then to verifying some of these areas for this coming year, and we're looking to see if we can fund that under the collaborative program under our funds this year. So, you know, I'd like to see some more samples from the Buckman diversion area. I'd like to see more samples from the Rio Chama area. We are talking about where are areas where we can truly say that we're going to get a negative hit, where are real true controls, and trying to expand that information as well. But yeah, if folks have some suggestions, let us know where we could sample.
Someplace scenic would be really good too.
I think it'd be interesting to go above Angostura Dam too. Have you done that? I'm looking and I don't see that.
We did not go above Angostura Dam. You know, we're very conscious of tribal sovereignty and really wanted to focus on more of the federal lands. Over.
Okay, there's some non-tribal lands up there, but yeah, I mean, it would be something, if we're talking about fish passage and things like that, it'd be good to get a sense for that, but I'm sure that would take some discussions with the tribes and pueblos.
Yeah, Denise, how far apart do samples need to be to sort of consider them to be independent of each other? So how
for each sample within that location?
For each sample within a location. So let's say we wanted to sample within the Albuquerque reach. How far apart are we trying to collect these samples?
Do you have an opinion on that? I would I would say space them out as far as you can.
Well,
and then take multiple replicates. So maybe if you could take a transect going downstream on one side, maybe then flip yourself over to the other side of the river and take a transect going down on that side and compare them.
I think you'd probably, so here you'd probably want to be, you know, as best informed by the ecology of the species as possible to identify, you know, like sort of suitability of different locations within that stretch of river, and then probably sample fairly densely in that reach, and then some other reaches as well, maybe not as densely. Because if you only have a small number of fish, there's not going to be a lot of DNA in the water. Yeah, I don't know if there's it'd probably be a trade-off between you know, just how much sampling you're capable, you know, that you have the potential to do in terms of, you know, manpower and costs and things would probably be a more effective determinant than trying to guess just how far apart they needed to be in the geographically.
There's no one size fits all answer yet.
Unless we want to do some pretty serious, you know, modeling of the eDNA production on a per fish basis and the fate and transport of that DNA in the particular types of system you're looking at.
Yeah, because I'm not sure, and maybe Mick can speak to this, and I see that we have a question too. But in terms of like fish sampling, when we actually going out there and doing fish sampling, how far apart is that sampling effort done? And maybe we would replicate that, for example, every 500 meters, every 1,000 meters, whatever it might be, and kind of replicate that with the eDNA and see how it correlates. I'm not sure. Let's go to Matt. I see your hand up.
Can you hear me?
Yes.
Yep.
Okay, sorry about that. I had a little trouble with that. Have you, maybe I missed this before also, but have you considered doing a sample further south, maybe below Elephant Butte, where the fish is extirpated, to not only take a look at some of those the potential of an area where there is no fish, but potential upstream contamination?
That's a interesting question, Matt. Yeah, we would presume that flow through Elephant Butte might be of sufficient time and duration to degrade the eDNA. So maybe something, maybe more of a transect approach, starting at the headwaters of Elephant Butte and going across the surface downstream and then sampling downstream of the dam as a first cut. And then if you're still detecting eDNA downstream of the spillway, then come up with a similar design to extend downstream. That's a really interesting question. Thank you.
Welcome.
I would note just real quick, when you guys do go back out to do some additional eDNA sampling, don't sample at the same time that you're taking your seining nets out there, because there's always going to be some residual DNA on those nets. We did have an issue with contamination with our first set of samples that came in. The non-target species were amplifying pretty regularly with the assays, and it turned out that there was just a little bit of Rio Grande Silvery Minnow DNA contaminating those samples just from a fin clip. So it does, eDNA sampling does require a pretty sterile method. So if you're going to eDNA sample, only eDNA sample, and don't be doing your traditional methods at the exact time at the same place.
We saw this with Asian carp work back in 2010 to 2014. We were getting eDNA samples from stretches of the Illinois River where they were also netting for big-headed silver carp, and the nets were being reused over and over again, and they were just absolutely covered with eDNA bearing materials, and so they were contaminating the sites every time they put the nets in the water, and they were taking their water samples at essentially the same time in the same area, and it was causing a lot of false positive kind of results. So you gotta think through all the little facets of sterile technique and potential contaminating behaviors or things that you can do that you need to avoid.
Okay, so 2 separate teams, eDNA samples first. Once they're off the river and down the road, then the other team can come in and mess around.
Yeah.
Good points. Okay, we've got about 7 more minutes, but I want to make sure that everyone's had an opportunity to ask questions. I see that there was a question in the chat. Was flathead chub included as a non-target species? And Mick answered, yes, we included flathead chub and the Rio Grande chub. And if you don't feel like speaking up here, you can put some more questions in the chat and I can read them out as well. Grace?
Yeah, sure. I mean, it is interesting, and I know we've been talking about some eDNA for a while. We know right now there's Rio Grande Silvery Minnow from Angostura down to Elephant Butte. And so I'm just curious what the proposal would be for use of eDNA in monitoring either genetics or presence, and whether you can get a sense. I don't think you can get a sense of the number of fish out there from eDNA. But so I'm just kind of curious what we would possibly use eDNA for, unless we thought we no longer had silvery minnow in the river and we couldn't find them from sampling. Would we sample, not find them, and then do eDNA? I'm just kind of curious what the use would be for this.
Well, there's focal sampling in areas where we think they might be using the area. We could look at that. I think one of the challenges with our traditional sampling is it's really good at catching young of year silvery minnows, but in years where we don't have a good spawn, it may start giving us some insights as to what the densities are, even though they're not showing up in the nets. That would be one direction to do some further investigation on.
And you can actually get some degree of abundance or biomass or rank abundance type of information from eDNA. You just have to, it just requires some additional work to look at the correlations between, correlations between biomass and eDNA signal strength. About half the papers that have been published on this, well, probably more than half, because there's a little bit of a bias to not publishing negative results, but you know, found positive correlations between amounts of eDNA and the biomass or numbers of the target species in the system. You just have to, again, you have to do some additional work to see how well how strong that correlation is. And there are ways like the paper that that was posted earlier that to sort of improve that correlation as well, looking at things like allometry and I guess the ecology of the eDNA, the load, you know, the fate transport of it. You know, there are various ways to you know, improve that, the strength of that relationship.
The other benefit of running that LOD LOQ with the synthetic DNA is developing a standard curve that you can then push back to your cycle threshold value that you're getting for each sample, where it's amplifying at, and then you can correlate that to an estimated number of copies of DNA for that sample that you detected. So like I said, the lower the CT value, the more DNA you're seeing in that sample. So that's very general and not modeled, but you know, the more DNA you're seeing, most likely you're seeing more individuals there, or you sampled closer to where that one individual was at.
Yeah, qPCR is quantitative, so it tells you how much DNA you've collected through the extraction process, and then, like I said, that sometimes correlates quite well to abundance. You have to, again, you have to kind of check it out and do some additional work there. We also have what's called digital PCR, which is another kind of quantitative PCR that's supposedly even more reliably quantitative, although I think we found that it performs about the same as our qPCR, but that's kind of the cutting edge for quantitative PCR kinds of approaches.
Thanks.
And I was just going to say, Grace, that and it could be used for some compliance and doing some work where, let's say you're going to put in a bridge in an area, so you can have a lot of earthwork in the river channel, and maybe you don't feel confident with some of your traditional sampling methods, and this could be another way to ensure that there are not any fish in the area when you begin your construction project. I also see that Andy Dean put in that it will be helpful for reintroduction efforts in the future. I'm guessing that that's because, you know, then we know where there are areas that they don't occur. Those would be maybe some places, or they have been extirpated through time. We could then go in there and reintroduce some fish. Thomas, I see that your hand is up.
Yeah, thank you. I was just going to agree with Grace, and maybe that point didn't get across fully. We also put a large number of hatchery fish into the river nearly every year. So I am also kind of wondering about how useful this is going to be within the Middle Rio Grande when we know that there's always going to be fish there. And in some years, there's 10 times as many hatchery fish as there are wild fish. And if you're not going to be able to distinguish between them, I'm not sure what you're gaining for just the traditional monitoring.
Yeah, and I do think, just adding to that, you know, I think it's worth looking at, just because if you're doing traditional monitoring, but maybe if people can grab samples and then they start getting a database that indicates where maybe you're finding more DNA, you know, more fish through DNA than not. Maybe we could do something with that. I just don't know yet, but I know if you're just trying to find out if there's a fish, we kind of know there's a fish. It's just a matter of, you know, how you're going to decide that, whether you're going to sample traditionally or do it through DNA. But lots of good information here. Thank you. It's really interesting.
Yeah, I mean, but we're also learning other things like, well, are they in the irrigation ditches, for example, you know, or where, and maybe even using this sampling method during certain times of the year, looking at floodplain versus river, or other ecological sort of data that can go along with this to make some conclusions about where they occur and when they occur there. But also, I mean, this is an endangered species, and there is always a possibility that these different populations will wink out, and this can help us track that and see if we can find, maybe there's going to be some sort of point where traditional sampling will not be as successful because there are so few of them in some areas. And so this could help us determine whether there's still some in there that we haven't found, right? If they still exist on the landscape in some areas, if there's some sort of refugia where they're being supported. But I think that there's some application now, but there's also potential for application in the future, depending on the trajectory of this fish. Okay, we're at 2 minutes. I see that Denise said if anyone has samples, fin clips, preserved tissue, that she'd love to test them and test the markers against them. So you can reach out to Mick Porter or me or Denise. Our emails are associated with the invite for this presentation, and we can look at getting those analyzed. Did I miss, before we leave, did I miss any hands up here or any outstanding questions? See anybody else with their hands up? Okay, well, thank you so much for attending this presentation. Thank you, Denise and Mick, for